Welcome, dear students. We will continue our lecture of physiology, lecture number 4. Today we will study the origin of action potential. Action potential is the activity which carries the impulse or the message or the signals which receives the cell and passes it to the next. And this is nothing but the action potential. Action potential is originated at the axon hillock. This is the axon hillock region, as we have seen previously. This is the dendrite, this is the axon, and this is the neck, or we can call it as axon hillock. This is the region or area where the action potential originates, or origin of action potential is nothing but the axon hillock, and receives from these dendrites and passes through the axon. Axon, correct. The action potential, as it propagates in the axon hillock, a wave of long signal passes through the axon and the currents flows inwards at a point on the axon during an action, and therefore this is nothing but the complete total action, and so called as action potential in a physiology. Potential means, or the potential spread out along the axon and depolarizes the adjacent section of its membrane. Now depolarization and repolarization will be studied in the next topic. Means movement of sodium and potassium ion, that we will see. Actually we have already studied the same mechanism in sodium potassium pump, but how it works, that we will see in the action potential. Now brain cells called as neurons sends information and instructions throughout the brain and body, or we can say give and take, or we can say to and fro mechanism of information is facilitated by the neurons or nerve cells through the action potential. How the information, we will see. The information is sent via electrochemical signals known as action potential. This is the simple definition. And that action potential travels down the length of the neuron, complete length of the neuron. These neurons are then triggered or stimulated to release the chemical messengers called as neurotransmitters. So neurotransmitters are the chemicals which are transmitted in between the cleft and facilitate the movement of message or action or signal. And this neurotransmitter helps to trigger the action potential. Means real action potential is being carried out by these neurotransmitters in the nearby cells or the target cells where the message to be sent, and so it helps to spread the signal all over the membrane or cell. In this way the action potential is facilitated. Now furthermore, the origin of action potential studied in the next as repolarization and depolarization. In last we have seen that the potential spreads along the axon and depolarizes the adjacent section. Depolarization means this one, depolarization. In suppose this is the nerve axon, or this is the nerve cell and this is the axon. So axon have different nodes, and in between these nodes or in the membrane of the axon, as soon as the sodium plus ions enters, the membrane gets depolarized, and as the sodium enters, to maintain the equilibrium, potassium moves out, and as soon as the potassium moves out or potassium ions moves out, it is called as repolarization. So this is the positive and negative, activity of movement of ions in and out is called as depolarization and repolarization. And collectively we can call it as a action potential, which begins at the axon hillock of the nerve cell or neuron. A neuron can receive input from other neurons, and if this input is strong enough, means message which received to the cell is strong enough, sends the signal to downstream neurons. Transmission of a transmission of a signal between neurons is generally carried by a chemical called a neurotransmitter that we have already studied. Means these are the responsible material for the facilitation of action potential in the nerve cell, that is neurotransmitter. Transmission of a signal within a neuron from the dendrite to the axon is carried by a brief reversal of the resting membrane potential, and this is called as action potential. You see a single definition. Again we can note here, transmission of signal to and from or from the dendrite to axon is nothing but the action potential. When neurotransmitter molecules binds to receptor cells or receptors located on neurons dendrites, the ion channels open. Means as soon as the ion channel opens, the ions enter. As soon as hyperpolarization occurs, it creates it stimulates the signal to open the ion channels for potassium, and then potassium molecule moves out. In this way, the excitatory synapse, which form either on the dendrite or at the axonal endings, this allows the positive ions to enter the neuron and results in depolarization. Particularly, this is the depolarization of membrane and a decrease in difference in voltage between the inside and outside the neuron. Means depolarization also facilitates the difference in voltage, which decreases, means drops down. A stimulus from a sensory cell or another neuron depolarizes the target neuron to which the message is to be sent, and to its the threshold potential becomes lowers or drops down. As you have seen that this normally at rest is minus 70, which drops down up to minus 55 millivolt, and this is nothing but the decrease the difference in voltage. So sodium plus ion channels in the axon hillock opens. As soon as this activity happens, the sodium channels again open at the axon hillock region and allows the positive ions to enter the cell, and this is the actual origin of the action potential. Means action potential begins actually here in axon hillock region. Next, the action potential is to be described in both unmyelinated axon and myelinated axon, and the initiation and conduction of the impulses or messages or stimulus to the axons is picturized here. You see, this is a simple unmyelinated axon and this is myelinated axon. Means axon which is enveloped by a sheath. We can call it as a Schwann sheath or myelinated sheath, and therefore this is also known as Schwann cell. This is also known as Schwann cell because this also works as a single simple cell, and in between these two cells there is a node called as node of Ranvier, and the actual activity of depolarization takes place at the nodes only, and therefore these nodes are the site of depolarization and these are the sites of repolarization. This is in a simple way, but this is a simple node, a piece of nerve which works, or piece of axon we can say, a node, and therefore this summarizes as a single piece. So single piece is working as node. Now, the initiation and conduction in axons. Now, we have seen origin, then axon hillock, then movement of sodium and potassium, and resting membrane potential lowers down as upgrades. So, in between these two, the initiation and conduction of axons, conduction of messages or signals in axons is to be studied in both, either in unmyelinated or myelinated. The speed of conduction of an action potential in unmyelinated axon is proportional to the square root of the axon diameter. Means axon diameter plays a key role in the conduction of action potential. Thus, invertebrates have large diameter axons for signals that needs to be propagated rapidly, and therefore the action potential takes place rapidly. Once the sodium channels opens, the neuron completely depolarizes. We have seen in previous diagram. From here it is clear that as soon as the sodium molecules or ions enters, the membrane get depolarizes. This is the simple way. Now we will go to the next. Once the sodium channel open, neuron completely depolarizes. This is the simple way. To a membrane potential of about plus 40 millivolts. We have seen in last minus 55 millivolt among or out of the 70 minus 70. Here the plus 40 millivolt is required for the movement of this sodium and completely depolarization. Now furthermore, the action potentials are considered an all or nothing event. All or nothing event. In that, once the threshold potential means beginning potential or at the very first potential is reached, the neuron always completely depolarizes. This is another or second way by which the depolarization takes place. Here we are highlighting the depolarization, means the movement of sodium ion inside the neuron or the membrane. Depolarization occurs or takes place or carried out. This is the simple way. And as soon as or once the depolarization is complete, the cell must now rest. Means in between depolarization and the next furthermore repolarization, a hyperpolarization is there, and that is nothing but the rest period or reset period. We can call it as a reset period. Means again setting is being taken place. It's a membrane, and at the same time voltage back to the resting potential. Means complete action potential causes or passes or works or shows this reset mechanism. To accomplish this whole task of action potential, the sodium plus ion channels closes and cannot be opened further. And this is the complete depolarization action in action potential. Now we will see the repolarization. But prior to that, this reset and hyperpolarization is to be studied. This is the hyperpolarization. Now we will see how the action potential works in between these two, repolarization and depolarization, that we will see here. In this slide, this begins means action potential. The neurons refractory period. Refractory period is a special period in the neuronal activity through the action potential mechanism and reset mechanism, in which it cannot produce another action potential. As soon as one is completed, it stops there because its sodium channel will not open again. Once it is completed, it stops and closes, and at the same time another channel opens. Sodium channel closes and voltage gated channel for potassium ion opens, because the equilibrium maintenance is the need of the time at the time. and this allows the potassium molecules or ions to leave the cell. As soon as sodium ion enters, potassium ion leaves. This is the simple mechanism. As the potassium ions leave the cell, the membrane potential once again become negative. Means this positive ions movement makes the membrane potential negative. We are already studied the positive and negative millivolt powers and positive and negative ions and their movement through the membrane potential, which changes from positive to negative. And this diffusion of potassium out of the cell actually hyperpolarizes. This is one more different thing to be explained here as a hyperpolarization. Means in between depolarization, reset and repolarization, this is one pause, we can call it as a hyperpolarization of the cell, and in that the membrane potential becomes more negative. Here we have seen the positive and going to become negative, and therefore hyperpolarization drops down and becomes negative. Then the cell's normal resting potential. Means hyperpolarization is responsible to drop down the resting potential. At this point, the sodium channels will return to their resting state. Means once the action of sodium enters potassium movement is completed, again the movement of sodium ions starts, and at this point the sodium channels return and again opens, means that they are ready to open again for the further action potential or further membrane potential if the response or the stimulus is received collectively or simultaneously. The membrane potential again exceeds the threshold potential. Threshold potential is the beginning potential, as I told you. Eventually, the extra potassium ions, if diffuses out of the cell through the potassium leakage channels, bringing the cell from its hyperpolarized state and back to its resting membrane potential. Means the sodium, as soon as the sodium channel closes, potassium opens. As soon as potassium opens, potassium ion moves down, and as soon as the complete action potential is carried out, hyperpolarization occurs, and then the hyperpolarized state, the resting membrane potential comes to its previous resting state and again ready for the further action of the nerve. Now, the action potential we have seen and observed in the nerve axon or membranes. Here in the twitch or graph, how this is being recorded or observed. So in this figure, you are observing that 1, 2, 3, 4 and 5 points. This is the very beginning point, this is the initiation point, this is the depolarization point, this is the repolarization point. In between that, this is the resting point, resetting point. This is the drops down more than the normal level is nothing but the hyperpolarization, and in final stage again it is become ready for the further action potential. Now in between these two, the beginning is the resting potential, or we can call it as a threshold potential. This is the threshold, this is the resting potential, and this is the threshold or beginning. I told you that at the very beginning, threshold means beginning. As soon as the excitation of nerve impulse stimulated for the impulse transmission or the signal transmission, the threshold excitation occurs here and depolarization occurs. This is the depolarization state. In third, the peak or the optimum limit of this action potential is here, and this is the plus 30 millivolt and this is minus 55 millivolt, that we have seen already in the previous discussion, and this is the minus 70 is the normal level. But once it is drops down below the normal level, it becomes hyperpolarized, and this is the depolarization, this is repolarization. In between depolarization and repolarization, there is a peak of action potential, and this is the whole picture of action potential. Now, we will see here a stimulus from the sensory cell or another neuron causes the target cell to depolarize towards the threshold potential. This is the stimulus received to the second. If the threshold of the excitation is reached, all the sodium channels open and the membrane depolarizes. This is the membrane. Sodium channels causes the depolarization and potassium channel causes the repolarization, that we have already studied here, sodium and potassium channels. Fourth, the membrane becomes hyperpolarized as potassium ions continue to leave the cell. Here, if the potassium ions continually moves down, it drops down and causes the hyperpolarization. And this is the last, where the potassium channels closes and sodium potassium transporter restore the resting potential, or we can call it as the equilibrium is maintained. This is the equilibrium maintenance level, and these are the steps of complete action potentials. Now the next topic or fifth topic is propagation in myelinated and non-myelinated nerve fibers. We have already studied the myelinated and non-myelinated nerve fibers, one having myelin sheath and another having no myelin sheath, that is myelinated and non-myelinated. Now, according to the type of membrane or according to the type of neuron and its membrane, the theory of nerve impulse movement, its propagation and passage of the waves of depolarization is different. According to this membrane theory, the nerve impulse is propagated and depolarization or repolarization is consequently observed or occur. But here in these nerve fibers, the depolarization is to be studied previously or priorly in priority. We will see how the propagation is there. First, when the fiber is excited at a point, means as soon as the excitation or stimulus receives the nerve fiber, the polarity is reserved, means at one pole the stimulus is received, then the polarity is maintained to pass the message through the axon, and this is reverse polarity is due to increased permeability of sodium ions to the membrane, and this develops depolarization waves. We have previously seen that the movement of sodium ion causes depolarization and movement of potassium recovers the repolarization. Secondly, a local circuit current flows between the depolarized membrane and the resting membrane areas. Next, positive current flows inwards through the depolarized membrane and outward through the resting membrane, and in this way circuit is completed. As previously we have seen that positive current flows and it membranes the resting membrane potentiality by movement of potassium and sodium ions, then the equilibrium is maintained and action potential is completely carried out. The local depolarization current then exits the adjacent portion of the membrane, progressively more and more depolarization. Means as the moment of maximum ions, the activity of depolarization is continually going on. The depolarization waves travels in all directions along the entire length of the nerve fiber, and in this way the actual action of depolarization takes place through the axon. And here we stop our today's lecture on the movement, propagation, carrier, action potential, depolarization, then reset, hyperpolarization and repolarization. So the repolarization in detail will be studied in the next topic. So today we are completing here our today's speech. Okay, see you to the next lecture.